our today's topic is regarding the development of green analytical method using UV visible spectroscopy. So when we are undergoing the research work, and if our area of choice or the method, whatever we have chosen is the UV visible spectroscopy method, how we have to assess the greenness of the method. And I just want to show you some 3 or 4 articles which have utilized this greenness assessment. That is by developing the method using UV visible spectroscopy. Yeah. Our first method is greenness assessment of UV stability spectrophotometric method for determination of dolphinpridine in presence of its oxidative derivative. So the author has developed an analytical method for determination of dolphinpridine in presence of its degradant, and the method chosen was the stability UV spectrophotometric method. And after development of the suitable analytical method, the greenness of the method was assessed. So that is the research work done, and this is the DOI of the article. And coming to the abstract, the abstract provided by the author is the study described 3 spectrophotometric techniques. So simultaneously, both the compounds, that is a drug and its degradant, was identified and determined by using 3 different spectrophotometric methods. So that is namely the derivative ratio method, ratio subtraction method, and bivariate method. And the required things were done for establishing the methods, and finally the method was validated based on the ICH requirements, and the parameters like accuracy, precision, and repeatability were also done, and it got the satisfactory results. And whatever the degradants produced, those were confirmed by using the different methods like TLC, IRN, proton NMR also. And after establishing a suitable analytical method, the author has done the greenness assessment by using the EcoScale analytical tool and also the AGRI tool. And the drug of choice for this method is dolphinpridine. This is a chemically 4-amino pyridine, and it is a potassium channel blocker and mainly used for treating the disorders related to the motor functions and for improving the walking in adults. Uh, so like that, this is a drug of choice. And the main thing regarding this dalfampridine is it is having a pyridine group in its structure, and it is susceptible to oxidation. So whenever it is exposed into oxygen, or when there is an oxidation condition, this pyridine can get easily degraded, and it will get converted into 4-amino 4-aminopyridine N oxide. So usually, yeah, there are more chances for getting oxidized. So the author wants to develop an analytical method where that oxidant or the degradant of this pyridine should not interfere in any way during its estimation. So the author has developed a method such that in presence of this derivative or in presence of this degradant also, the drug has to be analyzed. So this is the degradant produced on oxidation. So the other has developed a stability indicating analytical method, and also it is a quick, novel, susceptible, and selective also. And so here are the stock solutions and the oxidative derivative solutions prepared. So I just highlighted the things because for assessment of the greenness, we want to know how much amount of the compound or how much volume of the solvent that was utilized, and what are the different chemicals that are used in this method. So I'm not concentrating on how to optimize the method, or what are the trials that were done, and what is the exact procedure for the specific method, but we are, as our scope is regarding the greenness assessment, I am concentrating on the chemicals and reagents used and what are the experimental conditions. That's it. So here the stock solution is taken as 100 microgram per ml. So for this 10 mg of drug was taken and dissolved in 100 ml of methanol. So we have also, for calculating the volume of the solvent, so we need all these things. So here for preparing the standard solutions, 100 ml of methanol was utilized. And for preparing this oxidative derivative, 100 mg of a drug was taken and 30% hydrogen peroxide was taken and 6 hours reflex. So this is very, very important thing, the condition. 6 hours reflex was done using the methanol and 3 times the methanol was used for extraction. So here the volume of methanol used was totally 75 ml for extraction. And then finally it was made up to 100 ml. So totally 100 ml of ethanol was utilized for this preparation of this derivative. And also we should note the point that is reflux for 6 hours, heating for 6 hours. And then the first method is the ratio derivative method. So as I have said earlier, we are not concentrating on the method procedure in detail, but the solutions prepared were 0.01 to 0.14 mg per ml of dalfampridine and 0.01 to 0.4 mg per ml of dalfampridine derivatives were prepared, and they were scanned, lambda max was taken, and the further procedure was made. And also the next method is also same, the same solutions were prepared, but the method is the ratio subtraction method. And the third method is the bivariate method. So for this the methanol was taken for preparing the standard solutions and the derivative solutions, and the procedure was done. And finally the obtained procedure, the established method was applied for the laboratory prepared mixtures. So the mixture was prepared by combining dalfampridine and its derivative, and finally the methanol was used as a solvent here in 10 ml volumetric flask, and the solutions were prepared, and the same procedure, the same methods, the 3 methods were applied for the laboratory prepared mixtures also. And then so the same thing was applied for pharmaceutical preparation also. So whatever the tablets here, the 10 dalfrasosis tablets were taken, and the same procedure was applied by using the methanol as a solvent, and finally the volume was made with water. So different concentrations were prepared and it was analyzed. And these are the validation parameters done, and the statistical assessment was also done. So ratio derivative method, ratio subtraction method, bivariate method, and this is the already existing method. So just to compare, for comparison, the other has kept this one. So the mean obtained, the parameters mean got this value, and standard deviation and number of samples they took, and statistical evaluation, t-test and f-test were done. So they found very good results when compared with the previous method. And the other has given a conclusion that so it can be successfully applied for determination of dalfampridine in presence of its oxidative derivative or the degradant, and which is found to be a very sensitive and also a selective method, because it has to be estimated in presence of its derivative also. And finally the method was validated, and greenness assessment was also done. So, and this is the greenness assessment profile provided by the author. So as we all know about the agree tool, which is a software, so you just need to put all the things, 12 parameters, and if we give the inputs of any sample treatment, so here for preparing the oxidative derivative we have used the sample treatment. So reflux was done using the hydrogen peroxide, so that's why it is not completely green, so it is only yellow. So and then the sample amount, so very less a sample was taken, so it is to the 2 per second one is green. And device positioning, so this is not completely green, so it was like between the green to and yellow. And fourth one is sample preparation and stages, so very less amount of samples were used, so it is a green. And automation and miniaturization, so here there is nothing like direct taking sample from the field within the lab itself done, and so it is also a green. And derivatization is not there, just the extraction. And then waste, so we are not doing any waste recycling process, so it is a yellow. And analysis throughput, so it is not a highly speed analytical technique, so it is not completely green, so it is in between yellow to green. And orange, it's like orange. And then ninth one, energy consumption, so it is also not completely green, because we are using the reflex for 6 hours, so it is more energy consumption process, so it is not completely green also. And then 10, source of reagents, so whatever the reagents we are taking, those are not completely green. because we have utilized the hydrogen peroxide, which is having some disadvantages also. So this is it's, somewhat not green, so it is like a yellow color. And 11, toxicity. So it is also not completely green, because so we are using methanol, which is not a completely green solvent, but when compared with the other, methanol is the best one. And then the 12th one is operator safety, as the operators get exposed to hydrogen peroxide and also heating for more hours. So there is some risk to the operator. So it is also not a green. It is like light orange color. So like this, the method was assessed for different 12 parameters using the green, and it was found to be 0.65 only. So if it is more than 0.75, it will be considered as a very good green analytical method. So the other was found like, so it is 0.65. It is also a good green, but if it is more than 0.75, still it will be more good.